The final session of today, it's going to get away from genetics and studying the interplay between the tumor tissue, the local microenvironment, and also how the liver repairs itself and how this relates to cancer processes. And it's my pleasure to welcome my very close friend and great scientist, Steve Artandi from Stanford. He's a professor of medicine at Stanford and a medical oncologist. And he's an expert on the telomerase protein and has done very fundamental work both in the biochemistry of telomerase, but also its functions in both protecting the cells, but also new functions in stem cell biology, and that's what he's going to speak about today. So welcome, Steve.
Okay, well, I want to thank Nabil and Katie and the foundation, and thanks for inviting me for this wonderful meeting and allowing me to share our recent work on liver renewal. So just as a brief introduction, telomeres are the nuclear protein caps that protect chromosome ends. You can see a cap at the end of every chromosome, and these are important in cancer cells and in stem cells and cells that have a lot of proliferative potential. They need to be maintained by an enzyme complex called telomerase. Telomerase actually elongates these chromosome ends, and that allows the end to be properly protected. In the absence of this telomerase mechanism, or if it's impaired in some way, then the telomeres will shorten progressively. This occurs during aging. This occurs during tumor development in many cancers. And when that happens, it unleashes a DNA damage response that triggers replicative senescence or cell death. So that's why about 95% of cancers upregulate this enzyme. Now, it's a complicated enzyme. I'm only going to talk to you about one subunit, and then I'm not really going to talk to you about any aspects of telomerase function, except that TERT is the telomerase reverse transcriptase, and it's a critical protein component in the enzyme, and it is regulated at the level of gene transcription. So its promoter is very tightly controlled, and that'll be important for this talk. Now, we know from studying cohorts of individuals who have germline inactivating mutations in the telomerase pathway that there's a whole series of tissue failure phenotypes that ensue in these patients over time. And I'm just going to point out a few. One that's relevant for today's talk is liver cirrhosis, another is pulmonary fibrosis, and another is aplastic anemia. So it seems that when you impair this pathway, tissues ultimately tend to fail. Now, telomerase is also known for being very closely associated with cancer. That's been known for a couple of decades. And this is true for, as I said, 95% of human cancers. And also, TERT, when overexpressed in cells that don't have telomerase, can actually confer immortal growth properties. That's one critical thing that telomerase does, is confer immortal growth properties onto cancer cells. Now, for many years, we didn't know how telomerase was actually switched on in cancer. And then these really striking papers were published in 2013, one from Levi Garraway's group and one from the Kumar lab. And they found that there are non-coding mutations in the TERT promoter region, one of two sequence changes that actually confer activation upon that promoter. They're highly recurrent in melanoma, but that's not the only cancer that they're seen in. They're actually the most common non-coding mutations in all of cancer, and they occur in about 60 to 70% of cancers in melanomas, in gliomas, in liver cancers, and in bladder cancers, as well as some others like thyroid cancer and liposarcoma. So you can immediately see that too little telomerase is bad, leads to tissue failure, and then many cancers need to switch telomerase on through acquiring these activating mutations in the TERT promoter. So how do these work? This is something that we're working on in the lab, but I won't tell you about today. So there are one of these two mutations in the very proximal promoter. They create a new ETS family consensus binding site. ETS is a transcription factor binding. This seems to be the protein, the GABP, which is a heterodimer protein that seems to reside at this site and is critical for activating the TERT promoter. Okay, now this observation that people with inactivating mutations in the telomerase pathway are suffering tissue failure led me to think that, well, perhaps many or all of these tissues are actually maintained by subsets of telomerase positive cells that had not yet been discovered. And so I set forward a program in my laboratory many years ago to see if we could try to discover those cells prospectively. This turns out to be very hard because the TERT promoter is very weak, telomerase is expressed at very low levels, and so it's hard to identify which cells might express telomerase. But we succeeded initially by inserting a TD tomato, it's a relative of red fluorescent protein, into the TERT locus through homologous recombination in mouse ES cells, and we found that those ES cells were in fact red. We made mice with those ES cells, and well, first we showed that when we differentiate those ES cells, then the TERT promoter activity will go down as these cells differentiate over the course of this 20-day period. So in vitro, in these cells, telomerase levels are high, the TERT promoter activity is high, but as they differentiate and no longer renew, then that promoter activity goes down. When we made mice, we can look at a renewing tissue, for example, in the testis of the mouse, where there are well-understood stem and progenitor cell relationships, and we found that these red cells are the cells that have the highest TERT promoter activity. Those are synonymous with the professional spermatogonial stem cells. And when those cells differentiate to make their neighbors that are committed progenitors, they switch the promoter off. So in a real tissue, the TERT promoter is wired such that it's on in cells that are giving rise to renewal of the tissue, and then the promoter goes down as those cells differentiate. Okay. Well, now I want to talk to you about the liver, and this is our recent work. So the liver has these unusual capacities in that it's very slowly turning over during normal homeostasis, but it has this very large capacity to renew upon injury. And this is the myth of Prometheus that you might be very familiar with, and that is Prometheus gave fire to man, and he was punished by Zeus for eternity. He was chained to this rock and had his liver eaten by a falcon. And if he had not been a deity, then this treatment would have induced cirrhosis, because that is what actually causes cirrhosis, are these cycles of damage and renewal. So the question is, how does the liver do this? Okay, so you all know about the general structure of the liver and its blood supply. The minimal anatomic unit of the liver is the hepatic lobule, this hexagonal structure. You can see the two blood supplies that merge to make the hepatic sinusoids that bathe the hepatocytes and then drain into the central vein. Now, zooming in on this portal to central axis, which is often discussed, here are the two blood supplies, again, merging into the hepatic sinusoids. All the hepatocytes are in touch with blood from the hepatic sinusoids, and of course, there's a second circulatory system where bile salts and acids are secreted from the hepatocytes, and they merge into the bile ducts. And then that fluid eventually goes into the gallbladder. Okay, now there has been a discussion of hepatocytes from this portal to central axis in terms of dividing them into zones 1, 2, and 3. Now, how does the liver renew? Well, for many years, it was thought that the liver renewed on the basis of these cells that emerge from the junction of the bile duct epithelium and the hepatocytes, and these are called oval cells. And these were thought to be bipotential cells that can give rise to both hepatocytes and bile ducts. And that was the dominant theory for some time, until three labs, Stanger, Willenbring, and Grompe, published around the same time using powerful lineage tracing techniques that they could mark these cells, and these cells did not give rise to hepatocytes in vivo, and instead, new hepatocytes seems to derive from pre-existing hepatocytes. So this really turned this thinking on its head, and I think was a major breakthrough, but it left open the question of, well, where do new hepatocytes come from exactly? Now, just to remind you again, a lot of the focus has been on the zonation of hepatocytes, and it's been known that there's metabolic compartmentalization along this axis. So in zone 1, the hepatocytes are more involved in gluconeogenesis and ammonia detoxification, for example. In zone 3, the hepatocytes are more devoted to glutamine synthesis or xenobiotic metabolism. And my colleague Roel Nusse at Stanford published a paper a couple of years ago finding that the cells that immediately surround the central vein are receiving Wnt signals, and they have special repopulating activities. So he could see during homeostasis that there were new hepatocytes deriving from these hepatocytes receiving Wnt signals around the central vein. And I think that there have been a lot of competing theories over the years that there might be special hepatocytes around the portal triad or around the central vein, but really focused on this sort of anatomic localization of special hepatocytes at the poles. Now, this is important to try to know how the liver renews itself, because I'm very interested in what the cell of origin for liver tumors is, and it's hard to really answer that question without a really detailed understanding of how the liver normally renews itself. And so you can imagine that understanding how the liver renews itself normally might be informative as to which cells represent the cell of origin for hepatocellular carcinoma or cholangiocarcinoma. Okay, so the way that we set out to address this problem was we had the hypothesis that just like in the testis and just like ES cells, there may be special cells in the liver that have high telomerase, and that these cells might be the ones that are renewing the liver for the long term. And so we built a lineage tracing system where we inserted Cre-ER into the TERT locus. So Cre-ER is a fusion protein of the Cre recombinase and the estrogen receptor hormone binding domain. It's inert until the animal is treated with tamoxifen, which activates the protein post-translationally so that it can act on its target cognate LOX-P sequences, as shown here. So we build in a second transgene, that strong promoter, LOX-LOX, TD tomato. So all the cells in the animal are originally not red, and when we give a dose of tamoxifen now, we can make the TERT-positive cells red, and those cells, importantly, are labeled permanently. So the power of lineage tracing is twofold. One, you can give just a single dose of tamoxifen, mark your cells of interest in what we call a short trace, but then you can instead, or in parallel, give a single dose of tamoxifen and wait a long period of time and do a long trace. And in that case, you'll actually ask, is this cell a mother cell for daughter cells that are born during that long trace process? Okay, so this is the work of Shengda Lin, a terrific postdoc in the lab who built the system and did the liver work. And this is a stitched together, zoomed out view of what we find in the liver. So we find that there are rare red cells in the short trace experiment. This is 3 days after giving a single dose of tamoxifen. And then the question is, well, what are those cells? Well, these cells are always hepatocytes, as we've demonstrated here by double marking with HNF4A. And we don't see any marking in other compartments in the liver. So we don't see marking in the bile duct. We don't see marking of stellate cells or Kupffer cells or lymphocytes or endothelial cells. So they're exclusively hepatocytes. We can then use a way of using a virus to label and mark all of the hepatocytes with a GFP, and we can find that our red cells are about 3 to 5% of the total pool of hepatocytes, and then we can use this trick to isolate the cells by FACS. And now we just want to ask, do they indeed have higher telomerase? And that is actually true. They have about 5-fold higher telomerase by enzyme assay, about 10 to 12-fold higher TERT mRNA levels by qPCR. And when we actually isolate these cells and cytospin them onto glass slides, we can then do single molecule FISH for TERT mRNA, and we find, in fact, that the TERTHigh subpopulation has very high TERT message at the single molecule level. Now, if these are special cells, then if we do the long trace, we'll see that their lineage will expand over time. So this is just during normal homeostasis. What we do is we give a single dose of tamoxifen, and then we will, in many cohorts of mice, isolate mice after 3 days, 1 month, 2 months, 3 months, 6 months, 1 year. And you can see that there's this marked expansion of the TERT lineage over time. And what this is saying is that, in fact, these are very special repopulating cells that have much greater repopulating activity than their neighbors, and that's all quantified here. You can see this very linear expansion, and here's the oil control where no tamoxifen is given, and you see no labeled cells. So that really suggested that these are unique repopulating cells in the liver. Now, where are they? Well, if we stain for the marker of zone 3 with an antibody to the marker glutamine synthetase, that lights up zone 3 cells. And you can see that our cells initially are interspersed mostly in zones 1 and 2. And then as they repopulate the liver, presumably replacing cells that are naturally dying off during homeostasis, you can see that they really nicely repopulate zones 1 and 2, and they have increased contribution to zone 3 as well, which is quantified in this bar graph here. So they're really distributed throughout all zones, and they're not located to one of the poles of the liver, as had sometimes been proposed for liver regeneration. Now, it's very important in these kinds of studies to actually do clonal studies, clonal labeling. And so now we've, in this set of experiments, we've backed off the amount of tamoxifen, give about 12-fold lower TAM, so that we're marking very, very rare hepatocytes. And that allows you to actually understand what's happening at the single cell level. And what you can see is that this is after 1 month. We have clones of 1, 2, 4, or 8 cells. These are contiguous clones, so there's no migration of the cells away from the mother cell. They're forming a distinct clone. And we can, through confocal imaging, do 3D image reconstruction. You can nicely visualize the shape of the clones, and we can quantify those in many mice, as shown here. At the 3-day time point, we have almost exclusively single-cell clones, but then as these cells are called into action to repopulate the liver, you can see that there's clonal growth, and the clones are moving up in size over time. Now, importantly, there's no change in the overall number of the hepatocytes, of the TERTHigh hepatocytes, so there's no net loss or gain of the cells. This seems to be established in adulthood, and then at least over the course of a year, there's no net change in cell number, in clone number, just clone size. So we investigated this even more in more depth. So then we did clonal analysis and co-stain with GS to try to understand the relationship, positional relationship between the clones that we're studying and zone 3, which had previously been shown to harbor some repopulating cells. And what we found is that the clones are almost always separate from zone 3. So they're in zones 1 and 2. And that's what is shown here in red in these bar graphs. And this is clones of 1, 2, 3, 4, 5, 6, 7, or 8 cells. But then there's a smaller percentage of cells, about 10%, 5 or 10%, that either border, so we call these bordering clones because they're right up against zone 3, or even rarer are these crossing clones that seem to have a mixed phenotype of both non-zone 3 and zone 3, at least as defined by glutamine synthetase staining. So this is further evidence that at the clonal level, the regeneration in the liver that's occurring is occurring throughout all zones and largely in the bulk of the area, which really is zones 1 and 2. Now, does this location make sense? Well, to just check that, we just simply did Ki67 staining on either wild-type adult liver or our TERT-CRE-ER adult liver. As shown here in this brown color, these are Ki67 positive cells. You can see that they're quite rare. And we just developed a metrics, what we call the position index, that reflects its the relative position of this proliferating hepatocyte relative to the nearest portal vein and central vein, and then graphed all those data in this manner, in this histogram, with the position at 0 representing the position closest to the portal vein, or this position at 1.0 representing the position that's closest to the central vein. And you can see that there's very little favoring of one position or another. It seems like the proliferating cells are distributed throughout these zones. And that reflects the location of the TERT-positive cells and the clones that are deriving from the TERT-positive cells. Okay, so now how are these cells dividing? Well, one could imagine one of two mechanisms. They could divide like a stem cell and both renew and differentiate. And in that case, we might expect that a high telomerase cell would give rise to itself when it divides, so self-renew to give rise to another high telomerase cell, and give rise to a differentiated hepatocyte or a hepatocyte that has low telomerase. Or, in contrast, it could divide just by simple cell duplication, and in that case, in the clones, all the cells would be high telomerase. So we set out to test this idea, and the way we did it was to give a single dose of TAM again, and then isolate red cells, either at 3 days, 1 month, or 1 year, cytospin them, do single molecule mRNA fish for TERT mRNA, and ask what's the relative percentage of cells that have abundant TERT message. And here's an example of those data. So at 3 days, almost all the cells have many puncta of TERT message. That's also true at 1 month, and that's quantified here with this red marker means that about 80% of the cells have these abundant TERT mRNA puncta. But after 1 year, you can see that now the clones are comprised largely of cells that have low TERT. But importantly, there's still about 20% of cells that retain high TERT in those clones. And I think that this is really consistent with the self-renewal and differentiation model, where the TERTHigh cells are both renewing and differentiating to make an expanding clone. And so in considering these two models, it appears that it's the model on the left that is correct and that describes the behavior of these cells. Now, there's also been a lot of comment on ploidy of hepatocytes. So hepatocytes, when the liver is first made, are diploid, but then they become polyploid as the animal becomes mature. And what we did here is to actually isolate, after we've traced some cells, we isolate the TERT-high cells, TERT-low cells, and the TERT-high cells, and then look at DNA content by flow cytometry. And what we found is that there was really no difference between the TERT-high and the TERT-low. About 20% have a diploid DNA content, and then increasing percentages have a 4C, 4N content, or an 8C, 8N content. So there's no special ploidy that's associated with these cells. They look like bulk hepatocytes by that measure. Okay, so how are these cells different? Well, we set out to answer that question by doing RNA-seq. So we purified TERT-high and TERT-low cells and did RNA-seq. And this is a volcano plot that shows the results of genes that were expressed in a statistically different level. And you can see that there were many genes that were expressed differently. And over here are the TERT-high cells, genes that are different in the TERT-high cells. On the left are genes that were upregulated in the TERT-low cells. So the first thing we found in analyzing these data is that genes that are associated with proliferation are upregulated in the TERT-high cells. That makes sense based on everything I told you. But we were really very surprised to find that all ribosomal components were upregulated in the TERT-low cells, as were electron transport chain genes. These were also upregulated in the TERT-low cells, in the bulk cells. So this really led us to think that these are the worker bee differentiated hepatocytes. They're doing the heavy metabolic work of the liver in terms of metabolism, in terms of protein synthesis, whereas the TERT-high cells, the 3 or 4% of hepatocytes, those cells are more devoted to renewal and less invested in metabolism. And I think this makes sense if you think about the reactive oxygen species that is likely generated from all these metabolic activities. And in terms of chromosomal stability, it makes sense to maintain the liver with a subpopulation of cells that have high telomerase and also have low reactive oxygen species to minimize the frequency of mutations. This is also evident looking more broadly at the gene set enrichment analysis that derived. So in red are the TERT-high cells, in gray are the genes that are enriched in the TERT-low cells, and you can see that all of these fundamental processes that we think of differentiated hepatocytes executing, those genes are upregulated in the TERT-low cells compared to the TERT-high group. So we tested part of this just by simply giving EDU to mice, and those of you who've done these types of experiments in the liver know that there's very little turnover in the liver, so in order to get cells to incorporate EDU, you have to give the animals EDU in drinking water or through multiple injections. This is an example of giving EDU over 7 days in drinking water and then co-staining with the marker HNF4A, and you can see that the basal rate of proliferation or incorporation of EDU in this context is 1%, but that rises to 6% in the TERT-high cells. So this is further evidence that the TERT-high cells are much more proliferative than the bulk hepatocytes. Now, what about injury? I introduced this part of the talk saying that the liver has this very significant capacity to renew itself when injured. So we've done a couple of different injury models. One is to give the drug DDC, and DDC causes a global injury. And what we find here, this is the control in the mice that received a normal diet. So you see these rare cells that are the TERT-high cells and their progeny after 1 month. And here are 2 examples where we gave tamoxifen in 2 different ways. And after only 1 month, you can see that now about 35% of the cells are red. So this is equal or equivalent to the amount of turnover that occurs in the liver after 1 year of homeostasis, all compressed into only 1 month. So the TERT-high cells appear to be doing the bulk of the renewal in response to this global injury caused by DDC. Now, DDC is also known for inducing transdifferentiation of hepatocytes. So now some new cells appear that have the markers of bile duct epithelia. So we wondered if our TERT-high cells could give rise to such cells, and in fact, that is the case. So in this co-stain after 1 month of DDC injury, you see red, which marks the TERT tomato lineage, and green, which marks CK19, the marker of bile duct cells, and now we see this overlap. And this represents about 10% of total red cells after this 1 month injury protocol. Just to make it clear, when we don't injure and just do homeostasis, we never see bile duct cells that are red in this case, even after 1 year. So there is this plasticity in the liver, and this may be very relevant for cholangiocarcinoma. Okay, so there are other ways of injuring the liver. One is to give a single dose of carbon tetrachloride, and when one does that, this induces basically a chemical wound in the hepatocytes that surround the central vein. And so this is a histology at 3 days, which shows this necrosis around the central vein. And this heals very rapidly within 7 days. At the beginning of the experiment, we find that there are very few red cells, very few TERT-high cells in this zone 3, as I alluded to earlier. But after recovery from this injury at 7 days, you can see that now that there are many red cells in the area of the wound, where there's been recovery from the wound. So now the representation of the TERT lineage is almost 20% in that region that's healed from the wound. So the TERT-high cells, even though they're not commonly in zone 3, they can renew that zone, and they can then assume a zone 3 fate and look exactly like the original zone 3 markers in terms of cells, in terms of marker analysis. Okay, so it seems that the TERT-high cells are special and they have this exaggerated renewal capacity, but we wanted to know if they were actually required for renewing the liver. And this is a very difficult experiment to do. So we spent some time trying to develop a system for genetically ablating the TERT-high cells. And this is a system that has worked using AAV, adeno-associated virus. It's essentially a lock-stop-locks driving DTA with a hepatocyte-specific promoter up front, okay? So when we did lots of controls that I'm not showing you, when we infect the animals with this virus, there's no toxicity to the liver. If we co-infect with a Cree virus, that causes all the hepatocytes to die, and the animal dies within about 5 days. Okay, so now we're going to infect the TERT-Cree-ER mice with this virus, and then just simply give tamoxifen and count on the Cree-ER that's in the TERT-high cells to delete this lock-stop-locks element. And we were able to ablate about 80% of the TERT-high cells in this type of protocol. So we felt that was a sufficiently successful ablation that we could go on to study what happens in injury. So we did this experiment, and then we put the animals onto DDC diet to induce a global injury. And what we find is that there's this marked increase in fibrosis as measured by Sirius red, so increased collagen deposition in that case. And you can see here that this is a control where we did not deplete the cells, so you see this big expansion of the red lineage. Here that red lineage is reduced because we depleted the cells ahead of time. But what you also see is a strong activation of stellate cells, an expansion of activated stellate cells, the mesenchymal cells that help to lay down collagen. And this is commonly what's seen in fibrosis and in cirrhosis. So this is pretty good evidence that the TERT-high cells are required to renew the liver normally, and if you take those cells away and damage the liver, it's trying to renew, but it's an aberrant renewal process causing organ dysfunction. And also, a result that might be informative for cholangiocarcinoma is that when we do this experiment, we see a doubling of the cells that actually convert to the CK19 fate, to the bile duct fate. So when you don't have the professional renewing hepatocytes at work, then the non-TERT-high cells are transdifferentiating at a higher rate. Okay, so to summarize, what I've told you is we've identified this rare subpopulation of hepatocytes that express high telomerase. These cells are distributed throughout the lobule. They're not anatomically restricted to one pole, to the portal triad or to the central vein. And that's different from what everyone else has found. And I think that's important. So if we think about the organs that we know a little bit more about, like the gastrointestinal epithelium or the skin, those are thin-layered epithelia where you have the stem cells located at a point source in the tissue, either the basal layer of the skin or the crypt of the intestine, and there's net migration in the biology of those tissues, so that as the cells proliferate, they differentiate, and they're either pushed up the villus to be shed into the lumen, or they're pushed up the skin to be shed off the skin. And there's no such similar migration of cells in the liver, and the liver is a giant tissue. It's not a thin layered epithelium, it's a giant 2-kilogram tissue. So really, how can you renew it you really can't renew it using a similar design. And I think the way that nature solved that problem is by distributing these special renewing hepatocytes throughout the tissue. So we imagine that those hepatocytes can survey some volume in the hepatic lobule and sense signals that either come from a dying hepatocyte or emanate from the loss of that hepatocyte so that they can clonally expand to restore liver mass. And that's how renewal is accomplished in the liver. When injury occurs, then you see a marked expansion of those cells, so now marked acceleration of the clonal expansion. And if we experimentally deplete those cells, now when there's a need for new hepatocytes to be regenerated, that process is really failing, and instead you see this aberrant stellate cell activation phenomenon and collagen deposition that leads to a damaged liver. So I just want to thank everybody who did the work. This is the work of Shengda Lin, whom I introduced earlier. Chandresh Gajera helped build this mouse system, and Elizabeth Nascimento pioneered the clonal labeling studies. And again, I want to thank you for your attention. I'm happy to take any questions. Oh, yes. Sorry.